Cello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored the fundamentals of tumor immunology, how the immune system recognizes and attacks tumors, why cancer is so difficult to eliminate, and the clinical tools that have emerged from this knowledge. Today, we move deeper into the battlefield between the immune system and cancer in a fascinating process known as immunoediting and how tumors fight back through immune evasion. This concept is critical because it explains why some cancers are caught and destroyed while others grow unchecked despite an intact immune system. So let's talk science. What is immunoediting? Well, immunoediting describes the dynamic, ongoing interaction between the immune system and developing tumors. It's a two-sided coin. On one side, immune cells recognize and eliminate abnormal cells. On the other, tumor cells adapt to survive immune pressure. This process is traditionally divided into three phases. First, elimination. In this early phase, the immune system detects and destroys nascent tumor cells through immune surveillance. Cytotoxic T cells recognize tumor-specific antigens such as mutated p53 or abnormal HER2 presented on MHC class I molecules. NK cells step in to attack cells with reduced MHC class I expression. Macrophages and dendritic cells capture tumor antigens and stimulate adaptive immunity. Interferon gamma secretion boosts antigen presentation and anti-tumor activity. Many tumors never progress beyond this stage, silent victories we never notice. The second phase is equilibrium. When a few tumor cells survive elimination, they can enter a dormant state where their growth is restrained but not eradicated. In this Cold War, immune pressure limits expansion while cancer cells accumulate mutations such as alterations in beta-2 microglobulin that reduce MHC class I stability. Some cells begin expressing fewer immunogenic proteins, balancing on the edge between visibility and invisibility. This stage can last years or decades and can often be the source for late recurrences. The third phase, escape. Eventually, some tumor variants acquire traits that allow them to avoid immune recognition or actively suppress immune responses. For example, melanoma cells may downregulate MHC class I entirely to avoid cytotoxic T cell killing, but must also evade NK cells by upregulating self ligands like HLA-E to deliver inhibitory signals. Others overexpress PD-L1, shutting down T cell activity, or secrete immunosuppressive cytokines like TGF-beta to remodel the tumor microenvironment into a safe haven for cancer growth. Let's look at the mechanisms of immune evasion. When tumors reach the escape phase, they deploy a sophisticated arsenal to disable the immune system. First, antigen loss variants. Tumor cells can lose expression of key immunogenic proteins such as MART1 in melanoma or MUC1 in breast cancer, preventing cytotoxic T cell recognition. Many of these antigens are normally presented on MHC class I. Without them, cytotoxic T cells cannot see the tumor. Second, MHC class I downregulation, which is a common escape route that reduces cytotoxic T cell killing. However, low MHC class I expression makes cells susceptible to NK cells, which detect missing self. Tumors often counter this by expressing non-classical MHC molecules like HLA-G or HLA-E, delivering inhibitory signals to NK cells. Third, immunosuppressive cytokines. Secretion of TGF-beta, IL-10, and VEGF dampens T cell and NK cell function while recruiting regulatory T cells or Tregs. Fourth, checkpoint molecule overexpression. High PD-L1 expression engages PD-1 on T cells, switching them basically off. And fifth, metabolic and physical barriers. Tumors can create hypoxic, nutrient-poor environments rich in adenosine, suppressing immune function. Dense fibrotic stroma and abnormal vasculature physically limit immune infiltration. So, what is the clinical relevance of this? Well, understanding immunoediting is not just an academic exercise. It directly shapes how we diagnose, predict, and treat cancer. First, predicting the response. Tumors in the elimination or equilibrium phase often respond better to immunotherapy. For example, a high load of tumor-infiltrating lymphocytes, or TILs, in melanoma predict improved response to PD-1 inhibitors, while a high interferon gamma signature can signal ongoing immune engagement. Second, therapeutic targeting. Immune evasion pathways have become direct drug targets. PD-1, PD-L1 inhibitors, for example pembrolizumab and nivolumab, can release the brakes on T cells. TGF-beta inhibitors and stromal degrading agents aim to dismantle physical and biochemical barriers. Third, biomarkers of immune activity. PD-L1 levels can indicate likely benefit from checkpoint blockade, while loss of MHC class I components like beta-2 microglobulin may predict resistance. MSI-high tumors tend to respond better to checkpoint blockade due to higher mutational burden and more neoantigens. Fourth, cancer prevention. Immunoediting research supports early detection strategies. Liquid biopsies can detect tumor DNA and immune signatures before the full tumor escape occurs, opening a window for intervention. It is an evolutionary arms race. Immunoediting is essentially an evolutionary contest. Every immune attack exerts selective pressure, killing the most visible tumor cells and leaving behind stealthier variants. Over time, this can yield highly adapted, therapy-resistant cancers, unless we disrupt the cycle. To conclude, immunoediting shows that cancer is not merely uncontrolled cell growth. It's an ongoing duel between immune defense and tumor adaptation. Sometimes the immune system wins silently. Other times, tumors emerge from the shadows, armed with molecular tricks to suppress or avoid detection. By mapping these interactions, we can design phase-specific interventions, boosting immunity during elimination, breaking stalemates in equilibrium, and dismantling defenses in escape. In the future, integrating genomic, proteomic, and immune profiling will allow us to anticipate a tumor's next move and strike before it escapes. In our next chapter, we will move from this evolutionary chess match into the world of CAR T-cell therapy, where we arm immune cells with new weapons to hunt cancer in ways nature never intended. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.